Okay, everyone, so let's dive into the next set of lectures. So we will be focusing today on population genetics, what the genotype and allele frequencies are. We'll also mosey on over to inbreeding and talk about things like inbreeding depression. And then we'll kind of end with the Hardy-Weinberg equilibrium and assumptions and applications. And then once again, work on some more genetics practice problems. So, one of the most important concepts I want you all to know is the modern synthesis. Okay, so the modern synthesis, basically it's a time period between the 1940s, 50s, and 60s. And what they did was they combined Mendel's genetics with Darwin's and Wallace's idea of natural selection and evolution. So Mendel published his paper, and it was, he was an Austrian monk, and so it wasn't in English, and a lot of scientists didn't read it, so they didn't pick up on things right away. Well, Darwin and Wallace, again, they didn't, they had the idea of natural selection and evolution, but they didn't know the mechanism of what was actually being selected for or against. And so the modern synthesis was what actually combined these two ideas, so you should know that. Okay, and so basically what they do is they define biological evolution as changing allele frequencies in a population. So you're looking at how your allele frequencies change in your population over the generations. Okay, so also it added the idea of mutation, gene flow, and genetic drift as additional processes that could cause populations to evolve. Okay, so given the reading that I know all of you have done early, hint, read. Okay, so you should be able to pause lecture and then fill out the blanks. Okay, so take a moment, stop lecture, and then just fill in the blanks for all of this. Hopefully after you filled in the blanks, this is what you should have. Okay, so population genetics is the study of genetic variation within and among natural populations and the evolutionary processes that generate and maintain that variation. So it's really important to know to define a population as a group of individuals of the same species living in the same area that are capable of potentially interbreeding. Okay. And a gene pool is basically all of the alleles, okay, for all the genes in a population. Now, the way this differs is because we talked about diploid individuals, just having two alleles per individual, okay, if they're diploid. And if they're triploid, you got 3, and haploid, 1, okay? Well, in a population, you can have, tons and tons and tons of alleles, even though every individual that makes up that population only has 2. So basically what they did was they took the concept of alleles and genes and so forth, and they basically added a whole bunch of math, yay! And then they added it, they applied it to populations instead. So as we look at genetic variation within natural populations, you might be surprised to know some data. Okay, so fruit flies, I don't know if any of you have them flying around your kitchen right now. I did until I tossed away some old bananas. And I think I told you before, you can sometimes catch them and if you wish, and look at the color of their eyes, or maybe I'm the only one that does that. But anyway, when you're looking at fruit flies, typically they differ by their genetic sequence only by 1%. Okay, humans, on the other hand, differ only by 0.1%. All right. And so the way this came about is because of the historical population sizes. And we've talked about that a little bit with the wolves, if you'll recall, from the last lecture. All right, now we've talked about the sources of genetic variation. Okay, we've said that you can have mutation, you can have recombination. Oh, look, a place to define these. Okay, so what I'd like you to do for this moment is define the two of these, because that's going to become very important. So stop lecture, go ahead and define these, and then start up again. So as we're thinking about mutations and we're defining it, we know that this introduces new alleles. Okay. Additionally, we know that recombination basically makes recombinant genotypes. That should sound familiar, especially on an exam. All right, in which case it's not the same as either parental, because it's recombining and you're getting new combinations of genes that you didn't have before. And so you might be asking yourself, okay, how in the world do we measure genetic makeup of a population? And I would say, aha, that is an excellent question. Okay, so we do this 2 different ways. We measure what's called allele frequency. Okay, and that's the number of copies of a given allele divided by the total number of alleles of a gene that you're looking at. And this will tell you the probability of randomly drawing that allele from the population. Okay, so that's one way we measure it. Now the second way is through what's called genotypic or genotype frequency. And so that's the number of individuals of a given genotype divided by the total number of individuals. Okay, Hopefully this makes sense. If not, please let me know. Okay, everyone, it's time for us to actually apply this. Okay, so what I want you to do is assume wing development is controlled by a single gene, and the wingless allele is recessive. So the question is, what is the genotype frequency for the little w individuals? Okay, so what I want you to do is look at the population you've been given above and address the question. By the way, it's not C. So again, the question is asking what's the genotypic frequency of the wingless individuals, and hopefully you counted, okay, 1, 2, 3, 4, 5, 6, 7. We have 7, and we divide by the total number, which is 21, and that gives us 7 divided by 21. Okay, so let's try it from the other perspective. Okay, so reading through the question again, okay, it wants to know the genotype frequencies of big W, big W. Now, I'm going to give you a hint because things get more complicated, okay? So remember what they look like when you address this question. So this particular question is a bit tricky because it's asking big W, but the problem is winged is dominant. So if you just count up the individuals, you don't know who's big W homozygous, nor do you know who's heterozygous, okay? For this reason, the answer is C, cannot be determined, okay? That should make sense. And remember, you're just applying what we've already talked about. So this should kind of be, make pretty good sense. So let's say you have a population and you have an allele set for B, okay? And you know that you have big B, little b. So your genotypic frequencies will be big B, big B, big B, little b, little b, little b. And that's just, again, the number of individuals that have those genotypes. Now, you also can figure out allele frequencies, okay? So in other words, the number of big Bs divided by the total alleles, and then the number of little b's divided by the total alleles, okay? So hopefully you read this in the book. Now, I'm not super fond of the book. I keep telling you guys to do the reading. It's because it should present it to you in a different way that should make sense, okay? So the more ways you see something, the more likely something is actually going to click. So this is not my favorite part of the book, but still, it should give you some good information. Okay, so if everything going according to plan, you should realize in order to figure out the frequency of big B, individuals who are homozygous dominant have 2 copies of that. Okay, so you take those individuals times 2, then you got to add the heterozygotes because they have 1 copy. Okay, and then you take that divided by the total number of individuals times 2 because everybody's diploid and everybody has 2 alleles. So that is known as little p. Little p is always associated with the dominant allele. Okay. Little q, that is the frequency of little b. All right, so that's always associated with recessive. And when you do problems in this class, I want you to keep it that way. Little p is the homozygous dominant. Little q is the, little p is dominant. Little q is recessive. Okay, so if you're asked to find the frequency of little q, you would take little b, number of individuals who are homozygous recessive times 2 because they have 2 copies, plus the heterozygotes divided by the number of individuals, and there's a typo in there, by the way. Take that times 2 again. Okay, so it's total number of individuals times 2. Now the other thing I want you to get from this, and write this down now, little p plus little q should always equal 1. If they don't, there's a problem. Okay, my suggestion to you is to always calculate it by hand, because if it doesn't equal 1, you know you've got a mistake somewhere and you've got to fix it. Okay, some people choose to take the shortcut. If they find 1, then they just take 1 minus that, but then you better be right, and there better not be any mistakes, all right? So write little p plus little q always equals 1. Okay, so let's apply this. What I would like you to do is take the information that's given here, okay, and I want you to find the frequency of the big M allele, okay? So do as we showed you earlier, and then figure out the frequency of big M. Go ahead and pause lecture and work through the problem. So in order to figure this out, there's 2 ways you can do it. You can take the counting alleles method, where you have big M individuals times 2, plus the number of heterozygotes divided by the total number of individuals times 2, which gives us 1,200 divided by 2,000, or 0.6 or 60%. The other thing you can do, given the fact that the genotypic frequencies are calculated for you, you can take 0.4, okay, plus half, which comes from big M frequency, plus half of 0.4, which is like 0.4 divided by 2, because that's the heterozygotes. And then you take that and just add those up, and that also gives you 60%. Now, I honestly don't care which way you do it. Personally, I like the top method just because it's a nice way to count and make sure you don't make any mistakes. But that's just me. But again, if I ever ask you, actually I should say when I ask you to do a problem like this on the exam, show me your work, okay? Because if you just have a little tiny mistake, I can give you most of the credit. But if you give me the answer and it's completely wrong, I have nothing to work off then. So, okay, and so now it's time for us to focus on the genetic consequences of inbreeding. Okay, so technically the definition of inbreeding is mating between close relatives. Also in plants could be self-fertilization. And so, you know, from a human perspective, we think, ew, gross. And there's actually an evolutionary reason for that. Because if you have too much inbreeding among lineages, what will happen is you will increase the proportion of homozygous offspring that might have deleterious alleles, okay? And so, for example, let's say you have a plant that can only self-fertilize, okay? So each time, let's say this plant is a heterozygote. Well, each time it self-fertilizes, 25% of its offspring will be big B, big B. So remember, it's big B, little b crossed with big B, little b. Do the small Punnett square on the side, okay, just like they've done here. And you see that 25% of the offspring are big B, 50% are big B, little b, and 25% are little b, okay? And so this will happen every single time you cross a heterozygote with a heterozygote. So you're always reducing the number of heterozygous individuals by 50%. Okay, so now the frequency of the alleles, by the way, I want you to pay attention to this, doesn't change. The frequency, of course, is 25% plus 0.5 divided by 2 equals 0.5. And then for a little b, which, by the way, big B should be a little p, little b should be a little q. 0.25 plus half of 0.5 equals 0.5. So that doesn't necessarily change, okay, or it shouldn't change. But what happens is you lose the number of heterozygotes every single time, okay? From that point on, big B produces, you know, only breeds with big B, little b only breeds with little b because it's selfing, okay, and big B, little b only crosses with big B, little b. So given this information, okay, think through it. What are the F2 genotype and the allele frequencies going to be if you're given this cross? So what they're asking you to do is to take the previous slide where you're given the number of individuals, okay, for the F2, and they want you to figure out the F2 genotype frequencies and allele frequencies. So just add up what they tell you in the previous slide, all right, and then go ahead and write the answers here and figure out the genotype frequencies and allele frequencies. And then once you're ready, flip to the next slide. So remember, it's selfing, okay? So it's self-fertilization, which means once you have an individual that's A1A1, they only cross with itself, so it'll be A1A1. Once you have an individual that's A2A2, they cross with themselves, meaning it's only A2A2. And every time you have a heterozygote, once they, you know, cross with themselves, you get a 25%, 50%, 25%. And so over subsequent generations, if only selfing is allowed, the number of heterozygotes is going to drastically decrease, okay? So genotypic frequencies will change, but allele frequencies will not. And that should make sense. So again, just a reminder, inbreeding is mating between close relatives or self-fertilization. This will increase the proportion of homozygous offspring. And if only selfing is allowed, by the way, this actually does violate the whole idea of random mating, which is a condition for Hardy-Weinberg equilibrium, which we are going to dive into in the next couple of slides, okay? So in order to have Hardy-Weinberg, you cannot have inbreeding. Just keep that in the back of your mind until we keep going through the rest of the slides. If this doesn't make sense, definitely please let me know. Speaking of Hardy-Weinberg, it's time for us to go ahead and dive into that portion of lecture. So the first thing we're going to do is define it, talk about the assumptions, and then talk about why this is actually used in science today. So as we've discussed, population genetics focuses on the genetic makeup, so allele and genotype frequencies of a population and how those either change or don't change through time, okay? So we're going to focus on allele rather than phenotype or genotype frequency as how we keep track of evolution. And I guess this is different from what's in the book, okay? So for us, we're just going to focus on the alleles. Because again, we've said this before, you know, evolution is changing gene frequencies through time, and we're keeping that consistent. All right, so Hardy-Weinberg is basically suggesting that there is a stable allele frequency. So in other words, for that particular gene, there's no evolution. Okay, so in order for this to happen, all right, which by the way, most of the time in nature, you can imagine that this doesn't happen. Okay, so to maintain a population in Hardy-Weinberg is actually pretty rare. So in order for, to keep a population Hardy-Weinberg, that means you cannot have any evolutionary processes going on. No genetic drift, no gene flow, no mutation, no natural selection, no sexual selection. You can obviously see how most likely this is not going to be the case, okay? Because generally, all those evolutionary processes, that's what leads to changing allele frequencies through time. Okay, so what I want you to be able to do by the end of this lecture is to describe the effect that these 5 processes have on the levels of genetic variation and the allele frequencies for the trait that you're looking at. Okay, if you have any questions, definitely send me an email. Better yet, come see me in office hours. I like the whole Zoom thing because I actually get to see you guys. All right, so when we're going from Mendel to Hardy-Weinberg, okay, so when you consider a single cross, you use gametes produced by what? Additionally, when considering a population, you use gametes produced by what? Little p and little q, as we've said, are frequencies in this particular example of big A and little a alleles in the population. So your little p is homozygous dominant, the dominant allele, and little q is the recessive allele. So when you consider a single cross, use gametes produced by the mom and dad, okay? But when you consider a population, you're considering the gametes produced by everybody in the population, okay? So all members of that. Because remember, you can have all sorts of different forms of the alleles. So the Hardy-Weinberg principle was actually created independently by 2 different scientists. One by the name of George Hardy and the other by the name of Wilhelm Weinberg. And they created this in 1908, approximately. And basically the principle states that after 1 generation of random mating, okay, so the genotype frequencies for 2 alleles at 1 gene will be little p squared, 2pq, and little q squared. So remember, little p is the allele frequency of, the first allele, the dominant one I always use. Little q is the frequency of the second allele, okay? And we said earlier, little p plus little q always equals 1, okay? The other part of this formula that should look familiar is little p squared plus 2pq plus little q squared equals 1. Now, to the side of your slide, I want you to write a little p squared, and that's equal to big P. Okay, and that's a genotypic frequency of the homozygous dominant. 2pq, and you're going to write equals big H, okay, and that's the genotypic frequency of the heterozygotes. And little q squared equals big Q, okay, and that's the genotypic frequency of the homozygous recessive. So given this information, why is there a 2 in 2pq? So to address that question, you should remember that there's 2 different ways to have a heterozygous genotype. The A1 from mom and A2 from dad, as well as the A2 from mom and the A1 from dad. Okay, so that's why there's a 2 in front of the 2pq. And given what we talked about with heterozygotes, that should make sense. Now, given what you've been reading about Hardy-Weinberg, hint, hint, okay, you should be able to address this question. So blank and blank frequencies will remain constant in succeeding generations as long as certain conditions are met. Okay, so let's think through this and address those blanks. So hopefully if you thought through everything, you know that the answer to the blanks would be genotype and allele frequencies remain constant. Because remember, evolution, if Hardy-Weinberg equilibrium is happening, then evolution is not happening, okay? And so this is only true as certain conditions are met, and usually they're unrealistic and not what's going on in nature. So in order for Hardy-Weinberg to happen, okay, these are the unrealistic conditions that almost never happen in nature. The population size is very large and stays that way. Well, usually no. The population is closed, so there's no migration and no gene flow. No mutations. Mating is completely random, so there's no sexual selection, no inbreeding. And then all genotypes in the population have an equal chance of surviving and reproducing, so there's no natural selection at the gene under study. So if all of this is likely to not happen in nature, you might be asking yourself, why in the world do they still use this? And I would say, aha, that is an excellent question. So I want you to write this down. The reason we use this in science is we compare our data against Hardy-Weinberg as a baseline. Okay, and so if in fact, our population is in Hardy-Weinberg, that means that there isn't any selection going on and there's nothing interesting going on. Okay, and then we move on and answer another question, or we move on to a different set of genes. However, by comparing it, we can tell that most of the time that's not the case, and then we figure out, okay, why. All right, so Hardy-Weinberg is nothing more than a baseline to check our data against to make sure that our population is not in Hardy-Weinberg so we can figure out what more interesting things are going on. So we're going to change up the notation a little bit because, again, I want you to see how other researchers use these frequencies, okay? So instead of big A, little a, often they use A1 and A2, okay? A1 is the dominant allele, or it's just one of them if you're not sure which is dominant. A2 is the recessive or just the other allele. Genotypic frequency is just like I suggested to you, okay? So the little p squared is big P, okay? So little p times little p. The 2pq, or A1A2, is H, and the little q squared, otherwise known as A2A2, is big Q, okay? And once again, little p is the frequency of A1, and little q is the frequency of A2, and little p plus little q better equal 1 for a gene that has 2 alleles that you're interested in. Okay, so it's time for us to actually work out a problem. Okay, so once again, genetics problems, yay! Oh, come on guys, these are fun. So you have a population with a frequency of allele A1 is 0.2, okay, and A2 is 0.8. If your population is in Hardy-Weinberg, what are the expected frequencies of each genotype? So in other words, what's big P, what's 2pq, and what's big Q? Okay, so you've got everything you need here to figure this out. Stop lecture, okay, go ahead and work through this, and then start us up again when you're ready. Okay, so you know that little p squared is big P, so 0.2 times 0.2 is 0.04, it's 4%. 2pq, 2 times p times q, it's 32%. Big Q, 0.8 times 0.8 is 64%. Now, if your population was in Hardy-Weinberg equilibrium, this is the genotypic frequency you would expect to see. Write this down. If your population was in Hardy-Weinberg equilibrium, this is the genotypic frequency you would expect to see, okay? The other thing I want you to notice is big P plus big H plus big Q is always equal to 100% or 1. It is always equal to that, okay? That doesn't tell you if it's in Hardy-Weinberg. What tells you here, you're assuming your population's in Hardy-Weinberg, in which case you have your genotypic frequencies here. Now, if you want to know, let's say you've got some data and you're curious if your population is in Hardy-Weinberg, and you have the frequency of little p and little q, you calculate how many genotypes you have of each kind, then use little p and little q to figure out the expected frequency, and then you compare the two numbers, and that tells you if your population is Hardy-Weinberg. I'm definitely going to add some more problems for you guys to do. I keep forgetting to do this. I'm going to add some more problems for you guys to do, practice problems for genetics, because I think especially with this it helps a lot. Okay, once again, we have another problem here for you guys to work through. The Amish population, high incidence of polydactyly, single dominant allele. One population, 36% were born with this. If you assume Hardy-Weinberg, the frequency of the dominant allele in the population is what? Okay, so stop lecture, work through this. Okay, so the key to this problem, by the way, is that it's caused by a single dominant allele. 36% have polydactyly, which means they're the homozygous and they're also the heterozygotes, okay? So what you have to do is work backwards and then figure out the frequency of the homozygous recessive, okay? So if 36% are born with this, and again, it's dominant, that means that 64% are not, okay? And if you assume Hardy-Weinberg, that means 64% is equal to little q squared. The square root of that is 0.8. Okay, and we've said before that if everything goes the way it's supposed to, little p plus little q is equal to 1. Therefore, little p is going to be 1 minus 0.8, which is 0.2. Okay, I realize this takes a while to think through. Okay, and I swear the more practice you have, the more obvious this will be. And so that's why I'm going to keep adding practice problems. Okay, assuming there's no dog barking, let's try another problem. Okay, so read through this problem. All right, and then stop the lecture and work through it, okay? So you have everything you need to solve this problem. So just apply what we've done. You guys have got this. Okay, guys, so it's really important you guys know how to solve this problem, and I want you to write this down, okay? So in the paragraph above, they have given you the observed genotypic frequencies. Write that down. 0.57 is the frequency of big N, okay? 0.18 is the frequency of the heterozygote, And you know that the genotypic frequencies all add up to be 1, so then it's 1 minus those 2, and that gives you 0.25 as the frequency of big M, big M. Okay? That's your observed genotypic frequency. Then what you do is you use those numbers to figure out your allele frequency. And we did this on the couple slides ago. Super easy. Frequency of big N is 0.57 plus 0.18 divided by 2 because it's the heterozygote. It's only got one of them. And that gives you 0.66. And I just caught a typo on the next line. The frequency of big M should be 0.25. So cross out that 0.57 and stick in 0.25 plus 0.18 divided by 2, and that gives you 0.34. Okay? Then the way you solve this is you use that allele frequency to calculate if your population was in Hardy-Weinberg, what genotypic frequencies do you expect to see? Okay? So you know that the frequency of big N is 0.66. So if your population's in Hardy-Weinberg, it's 0.66 times 2, and that gives you the frequency of big N, big N. I don't care for this one because it's codominant. If you designate one as, little p and little q and all that stuff, just make sure you know what's what, okay, and stay consistent. Now, to figure out the heterozygote, it's 2 times little p times little q, or in this case, a frequency of m times n. So it's 2 times 0.66 times 0.34. That gives you 0.449. That is expected heterozygote if your population's in Hardy-Weinberg. Then last but not least, the expected frequency, okay, for your homozygote, if your population's in equilibrium for big M, is 0.34 squared, which is 0.115. All right? Now, this still doesn't tell you whether or not your population's in Hardy-Weinberg. What you have to do is compare your values between the observed and the expected. Observed for big N is 0.57. Expected, we saw 0.43. Observed, 0.18 for the heterozygotes. Okay, expected is 0.449. These numbers are not the same. Because they're not the same, it is not in Hardy-Weinberg. Okay, and just like this, when I ask you to do this on the next exam, I will make it very obvious just like this is. If they're the same, they're going to be practically dead on. Okay, in this situation, they're very different. It's not in Hardy-Weinberg. Hopefully that makes sense. My suggestion to you, by the way, to really feel comfortable with this is work this problem again, but change the frequencies. Okay, and so change the 0.57 and change the 0.18. Give yourself another one and work through it and then see how you feel about it. It just takes practice. Okay, so I'm telling you now, this is a tough problem. Okay, if you guys can solve for this, you can pass anything I toss at you. Okay, during the second exam. Stop. Read through this. Solve this. The only hint I'm going to give you for right now is I would solve it the exact same way I have told you to solve multiple sets of alleles in the past, okay? Take a moment, go through this, give it your best shot, and then go ahead and start up lecture again. So hopefully that hint was enough, okay, that once again, they're giving you observed frequencies here. So you have to take these numbers, and then you have to solve for little p and little q, okay? You do it for the A separately, and then you do it for the B separately. Then what happens is for the A, you calculate the expected frequency. Okay, so you go through, and if your population was in Hardy-Weinberg, then the frequency of big A should be 0.7 squared. Okay, and that's 0.49. And big A, 2 times 0.7 times 0.3, and then little a, da-da-da-da. Okay, does that observe the genotypic frequency? That's what you got to look above. Okay, then you do the same thing for the B. Okay, so you solve for little b and little p above. Now go ahead and solve it for the bottom. Okay, so given all this information, all right, given the question that is asking you, okay, the answer should be B. So remember, if you deviate from Hardy-Weinberg, because remember this is just a tool, okay, then what that means is there's something interesting that is going on with your gene. Okay, so Hardy-Weinberg basically gives scientists a null hypothesis, and I can tell you when I was doing my research, I did check against this, okay? There's a program that helps you go through and do that, and I was very happy when my population was not in Hardy-Weinberg. So when the measurements indicate that it deviates, then you can figure out why, okay? There's either mutations, selections, drift, something's going on, and so the Hardy-Weinberg does approach one gene at a time. You saw that from the last problem, okay? And hopefully this makes sense. If not, I will hold live office hours on Monday and we can chat some more if you need me.